Well, it's my pleasure and honor to visit you folks. Again, I had the pleasure of visiting Baylor a couple of months ago. The difference between Baylor and THI is still kind of lost on me, but it's a pleasure to meet with you. My many friends here. I had a lovely dinner last night with my longtime friend AJ. What I'm going to try to do, I'm going to deliver several messages. One of them, I'll go ahead and cut to the chase, is delivered for the early career people here, that cardiology today is different than it was 20 years ago, and it's going to be different 20 years from now. And so you should think about what the future looks like. We're going to talk about some science around HFpEF. And at the end, I'm going to try to convince two things. I'm going to try to convince you that there's never been a more exciting time to be in cardiology and to be a cardiovascular investigator. And then at the very end, I'm going to do something a little bit fun, something a little bit more playful. I hope you know that cardiovascular diseases are the number one killer of men and women around the world, and the challenge is only getting greater. It is continuing to expand. Many of us don't remember that 1 in 4 women in this country will die of heart disease. 1 in 4. Contrast that with breast cancer, which is 1 in 40. A woman is 10 times more likely to die of cardiovascular disease than breast cancer. It is far and away, in the two red lines here, it is far and away the number one expenditure. That in many ways, cardiovascular diseases are bankrupting our society, and it's not just us. It is, in fact, around the world. Indeed, it is a global challenge. Cardiovascular diseases don't recognize political boundaries. As I'll show you, the face of cardiovascular disease is different in different parts of the planet, but it is the number one killer of men and women around the world. And it is evolving. So a moment of philosophy, if I may. As physicians, we try to cure disease. We walk into the patient's room and we hope that we can cure their disease. I challenge you to tell me how many cardiovascular diseases we can cure. There must be 500 cardiovascular diseases. We can cure infected endocarditis, pericarditis sometimes, reentrant arrhythmias, but in the vast majority of times, we cannot cure the diseases that we are trying to work with. That's also true in much of medicine. We can't cure diabetes. We can't cure rheumatoid arthritis. So even though we hope to cure a disease, in fact, 99.9% of the time, we have to settle for the next best thing, which is to transform an acute disease that would kill you into a chronic disorder that you can manage for decades. That is the definition of success most of the time in our profession. When I was a medical student at Duke a long time ago, I sat in a big room as a first-year student and heard 2 lectures about a syndrome that was emerging in Northern California and in Haiti, where young men were having their immune system just shut down. They developed rare infections, rare cancers, and it was a death sentence. It was completely unknown. It was quite scary, and it was called the acquired immune deficiency syndrome. We had no idea what the cause was, but if you got it, you were dead. Now, as of 2017, 50% of people with HIV/AIDS are over 50 years of age. We haven't cured HIV/AIDS yet, but we have transformed it largely into a chronic disorder that can be managed for a long period of time. So how about cardiovascular disease? Let me cite a couple of examples. In the 1950s, way before my time, if you had a heart attack, what they used to do was plant you in a hospital bed, turn the lights down, give you morphine, and wait a month. They didn't even let you out of bed for 10 days, lest you rupture your injury. At that time, in the pre-CCU era, if you had a myocardial infarction and you were in the hospital for a month, there was a 30% chance you were not going home. In-hospital mortality was 30%. You may know that Dwight Eisenhower, when he was president, was playing golf in Denver, had a heart attack. They planted him in the hospital at the University of Colorado and watched him for a month because the notion was there's nothing you can do about it. The horse is out of the barn. You just have to track the natural history of the healing response. By the 1970s, with the advent of the coronary care unit and these things that we do all there, the in-hospital mortality, unbelievably, in 25 years was cut in half. By the 1990s, with antiplatelet therapy and so forth, it's been cut in half again. Now, do you know what in-hospital mortality in your hospital is for MI? It's 3%. It's been cut in half and cut in half and cut in half, 30 to 3. We haven't cured myocardial infarction. Sadly, some people still succumb to that, of course. But in many ways, we transformed it into a chronic disorder, just like HIV/AIDS, and that chronic disorder is called heart failure. So this is just one example. What about the larger picture of cardiovascular death in this country? When I'm on rounds with medical students, I'll sometimes ask them, has age-adjusted mortality for cardiovascular disease that's in the last 60 years, has it gone up or down, and by how much? Since you're eating, I won't hold you to this. The answer is it's gone down a spectacular 75%. We haven't cured cardiovascular diseases, but people now are surviving their tachyarrhythmias, their acute coronary syndromes. Now, why is that? Why is it? What accounts for this amazing success? Some of it, of course, are statins and drug-eluting stents, but the vast majority of it is issues around lifestyle, the importance of blood pressure and exercise and weight loss and diet, staying away from cigarettes. That has accounted for this amazing success. Now, here's that same curve. Notice how in the last 10 years it's going backwards. We almost crossed over with cancer and became number 2, but we're still number 1, sadly. And we are now backing up. In fact, it's been said that the Generation Z individuals are the first ones in human history who, statistically speaking, will live a shorter life than their parents, statistically speaking. Why is that? Why are we backing up? Now, nobody knows the answer, but I think many of us believe it has to do with something I'll focus on in a minute, and that is the global explosion of cardiometabolic stress from obesity. Now, the face of cardiovascular diseases is different in other parts of the planet. You can see here in the BRICS countries, the atherothrombotic manifestations are still on the rise. I have occasion to go to China regularly in my role as editor to deliver the message of a tsunami of cardiovascular disease that is descending on Asia, where 60% of the human race lives. One of the hospitals I visited did 28,000 PCIs this last year, if you can imagine. Okay, cardiovascular disease today is different than it was 10, 20, 50 years ago. I urge you to think about where it is going. You all have an amazing, illustrious 50-year career ahead of you. Think about where cardiovascular disease will be 10 years from now, 20 years from now. It'll be in a very different place. My friend and mentor, Eugene Braunwell, said to me once, the thrombocardiologist of the 20th century will be replaced by the diabetocardiologist of the 21st century. You're all aware of the global explosion. This is just in the United States, of obesity exploding everywhere around the world. And sadly, our species is evolving in this direction. And I'm not a native Texan, but I can tell you in Dallas, we do this pretty well. And it is not just in the U.S., it is around the world. Let me digress by pointing out that different populations, they harbor their adipose tissue in different places. People like me living in the West, we put our adipose tissue subcutaneously, where it is harmful, but not as harmful as when it is packed around your liver, around your pancreas, visceral adiposity. For example, in China, where obesity doesn't look like anything like this. Okay, this is North America, right? This is what we do here in the U.S. This is what East Asia looks like. That's what obesity looks like. It's altogether a different magnitude. Nevertheless, the prevalence of diabetes in China is 14%, the very same prevalence as in this country. So sadly, in different parts of the world, in the Far East and in Southeast Asia, the adipose tissue is packaged around your liver and your organs, where it is especially toxic. So it takes a lot less obesity to trigger the cardiometabolic effects that we all see in some people, in some populations, than in others. And Hypertension is another expanding issue. You can see this is just 2010. You can see hypertension even in Africa, in Far East and so forth. And here's an interesting study. The likelihood that a woman will die of a non-communicable disease. It used to be that communicable diseases, TB and all kinds of infections and so forth, was the number one killer around the world. Now the emergence of non-communicable diseases, you can see that where red is where it's bad. Okay, red is bad. This is women, and here's every country in the world. Here's India, for example. 47%. A woman is 47% of women will die of non-communicable diseases before the age of 80. However, they are making some progress, but they're not on track to achieve the World Health Organization goals. Here's what China looks like, a little bit better, improving again, but not on track to reach international goals. Here's where the USA is, but we're not making any progress. Here are the winners. This is for women again, Japan, South Korea, and so forth. Here are the ones that are sadly in last place. For men, the picture is about 10% worse. There's a lot more red here, you can see around the world. You can see in India, it's about 10 points higher. For men, there's some evidence of improvement, but not on track to achieve World Health Organization goals. China's a hair better. U.S. is over here, but not improving. Japan, again, is in the lead, and these countries over here. What are those non-communicable diseases? Those non-communicable diseases we're talking about are cardiovascular disease. Again, I'll say again, obesity is exploding around us. And whenever I sit on an airplane, I know you watch people parading to the back of the plane. I'm reminded of the way it looks in our country, and as I've said, it's around the world. And where obesity is where diabetes is emerging, type 2 diabetes. At risk of oversimplifying, obese people develop diabetes. 20 years later, they develop cardiovascular disease and they succumb to it. The vast majority of people with diabetes succumb to cardiovascular disease. So obesity leads to diabetes, leads to cardiovascular disease. If you take 500,000 people with type 2 diabetes, 27% of them have heart failure. What kind of heart failure do they have? They have heart failure with a preserved ejection fraction, HFpEF. You're aware that our toolbox to treat our HFrEF patients is replete with many, many powerful agents, including several that are quite new. Arnie's, Entresto, for example, SGLT2 inhibitors. We have resynchronization therapy, mechanical circulatory support, maybe vericiguat down the road, maybe omecamtiv mecarbil down the road. We have a lot that we can do for our HFrEF patients. We've got nothing for HFpEF. Now, I'm aware there's a Sitagliflozin rep over there. We had a nice chat with him. We can talk about that later. The role of SGLT2 inhibitors in HFpEF is, in my mind, there's the evidence currently supports a morbidity benefit, heart failure hospitalization. There is sadly no mortality benefit. And I can tell you as cardiologists, we're spoiled. We have drugs that make our patients live longer, statins, aspirins, beta blockers. We want our drugs to reduce morbidity and mortality. And sadly, right now, that's not the case so far for SGLT2 inhibitors. Well, maybe the next trial will show something different. I hope so. It's been said that HFpEF is the single greatest unmet need in cardiovascular medicine. That is going to be, in many ways, the future. We already have more HFpEF patients in this world than we have HFrEF already, and it's going up. Thankfully, HFrEF is going down, but HFpEF is going up. So why do we have nothing to treat our HFpEF patients? Why is it that there are no agents to treat these individuals? And it's not for want of trying. There have been dozens of trials. Every single one of them has failed in treating our HFpEF patients. And in my view, my personal view is that clinicians who know exactly what HFpEF looks like are often not trained to glean molecular mechanisms. Conversely, brilliant PhDs who are professional mechanism solvers often don't know what the clinical realities look like. There has been a failure of communication across this divide where, in my view, physicians are often intimidated by PhDs who start throwing around terms like CRISPR-Cas9 and iPSC, and I don't know what you're talking about. I got to go. and the PhDs are intimidated to think, well, you're running around saving lives. I don't want to take your time. And there has been a failure in this space. I was fortunate enough to recruit to my lab a board-certified cardiologist from Italy who wanted to do a PhD with us and who's working very hard on this currently, Gabriele Schiattarella, now in Berlin. And we sat in my office and had a conversation where we bemoaned the fact that we have nothing for our HFpEF patients, nothing. And the preclinical literature is polluted with preclinical models, animal models of HFpEF that simply are not what we see in our patients and what has been documented by epidemiology. There are, I shudder to count how many studies have been done on HFpEF in animals. It simply is not what we see in our patients. And so Gabriele and I, to his credit, he took on a bold project, and then we decided to do something very simple. Let's just make a mouse that looks like a HFpEF patient. Most patients with HFpEF have hypertension, and most are obese with metabolic syndrome. So we decided, we conceived a two-hit hypothesis that we'll make a mouse that's obese with diabetes and a mouse that's hypertensive. There are hundreds of millions of people running around the planet right now with obesity and hypertension. So our two-hit model was we decided to raise blood pressure about 40 millimeters of mercury, a modest increase, and expose animals to a high-fat diet, rendering them obese with metabolic syndrome. These two hits, and our hypothesis was that maybe there's HFpEF at the end. So I don't have time to show you all the data, but some animals we exposed to no hits at all, some to a metabolic hit only, some to a hypertensive mechanical hit only, and some to two hits. And Gabriele traced these animals out for 5 and 15 weeks, and for a year, he threw stones at this model and had a checklist of about a dozen different phenotypes that he wanted to see before we were willing to call this HFpEF. And I'll telescope that whole year and tell you that after a year, we decided that these animals have the comorbidities we see in our patients. They have the systolic and diastolic dysfunction we see in our HFpEF patients. They have bona fide heart failure, exercise intolerance, and pulmonary edema, and they have the cellular changes that we see in our patients. So at that point, now it's been 6 or 7 years now, we planted a flag in the ground and said this is HFpEF until proven otherwise. And so then we started to look for underlying mechanisms. And one of them I'll illustrate for you, something called the unfolded protein response. You may or may not be familiar with that term, ER stress. Turns out when a cell is sick, like a cardiac myocyte, it can't process proteins normally. They don't fold, they precipitate out, and it'll kill the cell. And so we all harbor an elegant response called the unfolded protein response to deal with that, or ER stress. It has 3 arms. So here, all these misfolded proteins activate these 3 arms of the unfolded protein response in all of us. If you think about it in a simplistic way, if you have too many proteins that you can't handle, the unfolded protein response says, hey, let's make fewer proteins. Let's start to make more chaperones to fold those proteins. Let's start to degrade them in the cytosol and in the endoplasmic reticulum. It's a beautiful orchestrated biological response. And if all of this fails, the cell will commit suicide to preserve the organism. It's called the unfolded protein response. Again, it has 3 arms, PERK, IRE1 alpha, and ATF6. I'll tell you that this one right here is the most evolutionarily conserved, maybe the most important, maybe not. For example, yeast only have this one arm. So what happens is an unfolded protein binds to BIP, pulls it off, activating this molecule called IRE1 alpha, which is an endonuclease that cleaves mRNA, including the one coding for X-box protein 1, leading to a molecule here called X-box protein 1, XBP1S spliced. And it turns out XBP1S is a powerful transcription factor that we'd actually studied a little bit before with a different postdoc. He found that this pathway is activated by myocardial infarction. That was a discovery. And he found that this transcription factor, which is unleashed, activates 4 enzymes in something called the hexosamine biosynthetic pathway, leading to a post-translational modification that I won't go into much detail about. Bottom line is that it is cardioprotective. So this pathway leads to a cardioprotective response. So Zhao, who's now is an associate professor in Los Angeles now, he linked the biology of the unfolded protein response and the biology of the hexosamine biosynthetic pathway in this paper here. So this XBP1S pathway is activated by myocardial infarction. It's activated by high blood pressure. It's activated by diabetes. It's activated by every disease we've ever tested until here. For the first time in HFpEF, it is deactivated. You can see that high-fat diet turns it on, L-name turns it on, but when they come together, it is turned off, measured multiple different ways. So this was very surprising. This had never been reported in the cardiovascular literature, that this pathway was dialed down below baseline. You can see if we isolate adult mouse reticulomyocytes, we see that as well. And here's our friend, that endonuclease is deactivated. So this is shocking, very surprising. And so what I did was I got on the phone with a friend of mine at Johns Hopkins, which is an institution where they do endomyocardial biopsies on all heart failure patients, everybody, HFrEF, HFpEF. And I asked my friends there, can you tell me, please, what XBP1S is doing in human beings with HFpEF? And they wrote back a few weeks later that it is suppressed in human beings with HFpEF relative to HFrEF. So that was our first reality check, that this model we got appears to recapitulate what we see in human beings, A, and B, that this XBP1S repression, in fact, is part of the picture. So the next question is, okay, if there's too little XBP1S, what happens if we put it back? So we did that. We engineered mice. If this doesn't make sense, don't worry. We put XBP1S only in the cardiac myocytes only, using a paradigm that if there's doxycycline in the drinking water, the transgene is not on at all, you can see. But when we remove doxycycline, we can turn it on. So we can restore that deficit of XBP1S. So Gabriele gave these animals HFpEF over the course of 5 weeks. And over the next 2 weeks, he stopped the doxycycline, restored that deficit, all while these comorbidities are still present. And he found that the diastolic dysfunction improved just in 2 weeks. The animals started to run again, their pulmonary edema resolved, and pathological markers improved. So it looks like this XBP1S deficit does play a role in this cardiometabolic HFpEF as seen in human beings. So then the question is, why is there too little XBP1S? What's going on there? I hope I've convinced you that we know very little about the biology of human HFpEF. But one thing we do know is obesity is a situation of systemic inflammation. Obese individuals have a chronic low-grade systemic inflammatory activation. That is something we've known for a long time. It is called metainflammation, metabolism-related inflammation, metainflammation. And one of the hallmarks of metainflammation is a molecule called inducible NO synthase, which is upregulated in the context of this thing called metainflammation. So we found a study that, this is not our work, this is another group, where they were studying obese liver, and they found the same thing. They found that XBP1S is down in obese liver, and INOS is up through the roof. It's tenfold increased relative to eNOS. I'll remind you that INOS generates 1,000 times more NO than does eNOS. So this upregulation leads to a flood of NO all over the place, such that many, many different proteins are covalently coupled with this NO, something that's called S-nitrosylation. Dozens and dozens and dozens of proteins here are S-nitrosylated, including our buddy here, IRE1 alpha. So these investigators posited that this surfeit of NO leads to the covalent coupling and crippling of IRE1 alpha such that it can't do its splicing work. So we thought maybe that's going on in our model. And sure enough, we found that INOS is up 7-fold in these parts. If we isolate adult mouse reticulomyocytes, it is upregulated. There's so much NO flying around that many, many different proteins are S-nitrosylated, including IRE1 alpha. So here I get on the phone again with Johns Hopkins and say, can you tell me what INOS is doing in human beings with HFpEF? And they wrote back a few weeks later that it's increased 7-fold in human beings in endomyocardial biopsy specimens in humans with HFpEF. Now, you may know that some of the failed trials in the past on HFpEF actually gave the individuals NO donors like sildenafil or nitrates or beet juice to raise NO, because the notion was that there was a deficit in NO. And actually, if you look at those trials carefully, not only did giving them NO not make them better, it actually made them worse. And I think now we have a biological explanation for why that is. So what happens if we block that iNOS that goes up sevenfold? Here's a molecule that is specific for that. So Gabriele gave these animals HFpEF and it lowered their NO levels by blocking this iNOS enzyme. You can see it works just fine. And the animals, their diastolic dysfunction improved. The animals start to run again in only 3 days. So quite surprising. So here's where this model is, where the concomitant presence of mechanical stress, hypertension, and metabolic stress, obesity, leads to a phenotype called metainflammation that ultimately provokes HFpEF. We discovered this in mice and in rats, and we corroborated it in human beings. And I'm happy to tell you that this model, this so-called two-hit model now, has exploded around the world. There must be 300 or 400 labs, both in industry and in academia, who are using this. And I'm gratified because the HFpEF field was stagnant for 20 years. And now I'm happy to say that it's moving again. So then a little more science. How is it that XBP1S is a brake on HFpEF? I won't show you the data. Suffice it to say that XBP1S, remember that's a transcription factor, promotes the expression of a ubiquitin ligase, which degrades proteins like this, including this bad actor called FOXO1, which does a lot of mischief in the heart. Now, let me remind you that the heart is an omnivore. The heart has to consume substrate 24/7. Its preferred substrate is fatty acids, but it'll also eat amino acids, ketone bodies, glucose. It has to consume something. You know, the heart is only about this big, and it generates 6 kilograms of ATP every day, 6 kilograms. And the ATP gas tank in a cardiac myocyte only has enough ATP for 10 heartbeats. So if the cell stopped eating, it would beat 10 times and it would stop. So it has to consume all the time. Now, it turns out that the heart will pivot among its different substrates. If you expose the heart to hypertension, it'll tend to use more glucose. It wants to eat more glucose, but it can't eat glucose if it has diabetes. It's insulin resistant. So these poor hearts are being pulled in 2 different directions. They're being told to do something and they can't do it. And in fact, if you look at their mitochondria, you can see that they are structurally abnormal and ultrastructurally abnormal. The mitochondria are quite ill. And again, I'll telescope a long story, but this is a schematic of a mitochondrion, where we find that in HFpEF, the TCA cycle is normal. The electron transport chain is normal, but we've uncovered a defect in the intake of pyruvate into the TCA cycle, and 2 enzymes here in beta oxidation activation that are deactivated. They're hyperacetylated, thereby crippling their activity. And it turns out that the extra acetyl on there, the deacetylase that normally would cut that off, SIRT3 is downregulated, and its cofactor NAD is down. And so, again, I'm telescoping this story that our model of two-hit HFpEF leads to a hyperacetylation mitochondrial dysfunction response. And if we restore this NAD, either pharmacologically or by promoting its biosynthesis, the NAD levels go back up, the hyperacetylation response goes down, and mitochondrial function improves. So we have our first in-human clinical trial underway now at UT Southwestern, treating HFpEF patients with a NAD repleting strategy. Now, why the seeming predominance of HFpEF in females? It is true there's more HFpEF in females, and so we hypothesized that maybe female mice would be predisposed to this more than males. And so we compared them, exposing them all to the same paradigms. And in fact, we found that diastolic dysfunction was actually a little less in the females. It was there. The hypertrophic response was a little less. The LV remodeling index was a little less. It certainly wasn't worse. The animals, both of them had an exercise intolerance, but there was more pulmonary edema in the males than in the females, not the other way around. If we take these animals out to 15 weeks, the males continue to get worse, whereas the females plateau. So contrary to our hypothesis, it looks like the females, if anything, are a little bit protected. They still develop HFpEF, but it's a little bit less prevalent. And molecular markers, again, they're worse in the males than they are in the females. So that led us to the conclusion that there is no biological basis for why females have more HFpEF. So then what is the answer? And I'll tell you what many of us believe. One thing is females have a higher ejection fraction than males. That's true for people. That's true for large animals. It's true for mice, because the females typically are a little bit smaller, but yet they have to generate the same cardiac output. So they have a smaller heart generating, 5 liters of cardiac output. And so their ejection fraction is a little higher. And I believe that a woman who's running around with an EF of 65 and it drops to 55, that we would call HFpEF because it's north of 50, but in fact it's probably HFrEF, and that we're misclassifying some of these individuals. And so if you think about it, it seems ridiculous that we would use the same number for men and women, young and old, diabetes all around the world. And so there's a huge movement now to be, more granular precision use of that threshold going forward, which certainly makes sense. Now, most patients with HFpEF will encounter atrial fibrillation. Those two disorders travel together and they make each other worse. HFpEF patients can't tolerate AFib, the loss of the atrial kick or the activated ventricular response, and atrial fibrillation will worsen their HFpEF. So these two disorders that travel together actually exacerbate each other. So we asked the question: do these animals have a predisposition to atrial fibrillation? You may know that mice don't develop spontaneous atrial fibrillation. Their atrium is so small you have to do transesophageal pacing them into it. So if you pace the hearts of a normal mouse, we spread it out here, spread it a little bit more, you can see that they retain normal sinus rhythm. However, these animals develop atrial fibrillation, as you can see here. If we quantify that, the frequency of their developing atrial fibrillation and their duration is increased. So these animals do have a predisposition for atrial fibrillation, just like people. So then what is the mechanism underlying that? We looked first at the usual players, the things that we see in our HFrEF patients with atrial fibrillation, but we didn't find atrial hypertrophy as we would see in HFrEF. Then we looked for fibrosis, and we didn't find fibrosis either, suggesting that HFpEF-associated AFib has distinct mechanistic features relative to HFrEF. And in fact, what we have found is that this molecule called AMP kinase, phospho-AMP kinase, means it's activated. This molecule governs electrotonic coupling between myocytes so that the electricity travels between them. And when it is deactivated, they don't communicate their electricity signals normally. And we found that same thing. This is human beings with HFpEF. We're saying that same thing. So then we asked the question, what if we restore the AMP kinase by using a molecule we're all very familiar with called metformin? And in fact, the predisposition to atrial fibrillation diminished. And so this woman, Amanda, who is now has her own lab at UT Southwestern, is pursuing this aggressively on her own. So the future, where do I think things are going? This is my own personal opinion. This is what heart failure looks like in 2024. Heart failure is a syndrome, not a disease. It's a syndrome that's caused by hundreds of different diseases. We put them all together in a bin and call it heart failure. I dream that one day we will have disease-specific mechanisms for HFrEF, which we don't have currently. A precision approach that the different diseases that actually cause HFrEF can be treated specifically. Nowadays, you know this, that atherogenesis starts in childhood, in the second grade. Not because kids are developing fatty streaks in their coronary arteries, but rather they're learning not to take care of themselves. They're learning to eat badly. They're learning not to exercise. In the neighborhood I live in, kids ride these electric bicycles rather than a regular bike. My wife just shakes her fist every time she sees kids on these electric bicycles. They're learning not to exercise. They're learning not to take care of themselves. Heaven forbid that they learn later on to smoke. And so tackling these issues in childhood is going to be something much in the future in a term called primordial prevention, where we're trying to target the social and economic policies that affect health. We'll hear that term a lot more appropriately going forward. Genetics and genomics, we do this all the time. Polygenic risk scores, you're aware that every single one in this room is predisposed a little bit toward or a little bit away from cardiovascular disease based on the genes we were all born with, something we can't control. And so there's a lot of effort to try to figure that out by looking at polygenes, many, many different genes, to try to understand where are people pivoted to be protected or predisposed. There's much research on this. Of course, doing it in a population, a white population in the U.S. may not be generalizable to Black patients or to Asian patients or whatever. So there's a lot of work around trying to understand the generalizability around that. Machine learning and artificial intelligence. We just talked about that a second ago with Professor Heinrich, Professor Tegmeyer. Sorry, it is changing everything we do. It's already much prevalent in cardiovascular diseases in terms of interpretations of EKGs and chest X-rays, mammograms, and we'll see a lot more of this permeating everything we do. And as an editor, we struggle with this on a regular basis. How does it impact authors? Are authors using this inappropriately? Are reviewers using it? What are the challenges around that? It's an evolving area. The blockchain has been said to be one of the most transformative technologies since the internet. It is a distributed, encrypted way in which all of human knowledge is available on every single cell phone in this room on the blockchain. It is changing the way we interact with our science and with our patients and our world. Wearable technology, many of us have these devices to track our heart rate or our glucose levels or whatever. iPS cell technology, I'm sure you know about this. You can take a cell and back it up, turning it into a stem cell, and then say, okay, I want you to become a heart cell or a liver cell or whatever. That's called iPS cell technology. The Nobel Prize for this was given out several years ago. So this afternoon, you could see two patients in clinic. You could see Mr. Jones and Mr. Smith. Take a sample of blood, take the white cells, back them up into iPS cells, and then tell them how you want to become cardiac myocytes. Now you have Mr. Smith's cardiac myocytes in a dish and Mr. Jones's cardiac myocytes in a dish, and you can do clinical trials in a dish. You can test for toxicity, QT prolongation. Again, a major step toward precision medicine, which is something that we lag far behind in cardiology, right? Infectious diseases and cancer, they sample the tissue of interest. They grow the bug or they sample the tumor, and they can make the intervention precise. Whereas we can't because we don't routinely sample the myocardium. But this, I think, will begin to change that, such that we will move from what we do now, which is treating populations, to treating individuals. This will require sophisticated omics technologies, pharmacogenomics, epigenomics, proteomics, and so forth. But I think the harvest will be robust with new insights, new therapies, and new subphenotypes. Inflammation. I will submit to you that another place where the puck is going is inflammation. There have been many, many trials that have failed in this space. Steroids for myocardial infarction, that went away a long time ago. Anti-TNF alpha, many trials, nothing worked. And that all changed with the CANTOS trial in 2017, where for the first time ever, a surgical strike on a single inflammatory pathway, the interleukin-1 beta pathway, led to mortality benefits. That was like a thunderclap across the field. Inflammation pivoted from being a theoretical construct to being a bona fide mechanism that is targetable. And now we've had other trials that I don't have, Ladoco-2, Colcut, and others have shown this now in this pathway. This is that surgical strike pathway. Starts with something called the NLRP3 inflammasome, on now to IL-18, all this stuff. And you can see on the right here, what I have listed are the therapies that have been shown to provide benefit. On the left are ones that are currently being investigated. So this pathway is clearly a mechanism that contributes to atherogenesis. And again, I don't have time to tell you this story, but there are about 4 or 5 other pathways that are coming. And so inflammation is becoming something that will, I think, dictate the future of our world much going forward. CRISPR-Cas9, you're familiar with the notion that you can now edit the genome. We all have 23,000 genes, and The notion is that you can go after just one of them with this technology. There's a company that is doing this now in Boston where they targeted PCSK9. And you can see this is not in humans, this is in monkeys. You can see that they call it one and done, a single injection. PCSK9 levels drop to the floor, as does your LDL cholesterol. Now, this has been done now in human beings, reported at the AHA 6 months ago. Not in healthy, these are healthy monkeys, but this is in individuals with end-stage ischemic cardiomyopathy. And their PCSK9 levels drop to the floor and the LDL levels drop similarly. So this group posits they're targeting first people with homozygous FH because you can't treat them with statins if they don't make LDL receptors. So by targeting PCSK9, some of them have a residual, a little bit of LDL receptor that can go back up again. And those poor souls, as you know, they will develop myocardial infarctions in their 20s. And so the notion is that they might be willing, rather than having ischemic heart disease in your early 20s, to take a chance that this technology will target that one gene and not the other 22,999. They'll just hit that one. We'll see if their bet is true. They also have a second arm of this work where they're targeting angiopoietin like 3 for similar reasons. There's another company that's developing a targeting transthyretin for patients with amyloid. So the notion of editing the human gene pool is here. It is here now. It is happening today. Telehealth, of course, is something we all do a lot. Xenotransplantation is much in the news these days. I just read that yesterday. The individual who received a pig heart at the MGH was discharged home 2 weeks after receiving a pig kidney. Went home from the MGH yesterday. Now, the question is, why would you do pigs? Why is it always pigs? First of all, pigs are about the size of a human being. They grow really fast, and they have litters of 8 or 10 piglets, whereas monkeys are smaller and only have one. And so that's why all the interest is in the pig, and people are busily editing the pig genome to render it humanized so that you can do kidneys and hearts and so forth. And again, this is here now. You're aware that 2 years ago, the first human transplant of a pig heart occurred in Maryland. A second one occurred a few months later. Clonal hematopoiesis of indeterminate potential. I hope you've heard of this, called CHIP. All of us harbor hematopoietic stem cells circulating in our bloodstream that are constantly dividing, making new blood cells. But every once in a while, one of those clones will have a mutation, and it'll grow a little bit faster than its neighbors and begin to overtake the population. That's called clonal hematopoiesis. It's called indeterminate potential because sometimes it can lead to cancer, usually not. But what it does lead to is atherosclerosis. So by the time you reach 70, 10% of people over 70 have CHIP. By the time you reach like 75 or 80, it's like 30%. So it is very prevalent, and it is another mechanism that's just emerging, participating in atherogenesis. The metabolome, the metabiome. So you're all aware that each one of us has 3 kilograms of other organisms in our gut, 3 kilograms. And each one of us has a different spectrum of bugs, depending on what you eat, where you live, who you live with, do you have a pet, many, many things. And I can tell you that when the heart suffers pathological stress, it knows what's living in the gut. They talk to each other bidirectionally. That's absolutely a fascinating thing, that somehow, if you have a myocardial infarction, what is harbored in your gut impacts the extent to which you are susceptible to that. The biology is really fascinating and emerging rapidly. Social determinants of health. Again, I've already mentioned primordial prevention, dealing with the ways in which our society provides health care and education and food security is something that we are challenged with here. It's a global scourge. It's very different in other parts of the world, but it is a challenge around the world. So I'll stop before I do the fun part with something that's really out there, and that is there is a company in Northern California that called Altos Labs that is filled with really, really brilliant investigators and they are funded to a very, very great extent. And they believe that there's no reason why the human body should peter out at 80 or 90 years. They think that human beings can live to be healthy for 150, 200 years. We'll see if that's true or not. It sounds like science fiction, but these are very smart people who have a lot of money to work on this. So opportunities and challenges. The challenges have evolved. I hope I've convinced you that the challenges have evolved. At the same time, the tools at our disposal are absolutely robust and exciting. There's never been a more exciting time to be in this space. We as providers will continue to take care of cardiovascular disease as it evolves and as it expands. But the ultimate solution is going to be at the level of research, where we try to understand mechanisms and turn off this spigot. Okay, here's my fun part. One Saturday afternoon, I was sitting in my office, and I decided, I'm just going to say what I think about some of the keys to being successful in the early stages of your life. And I'd be interested in your feedback if you agree with me or not, but these are my thoughts. This does make me feel pretty old when I give this talk here. Maxim number 1, write. You know what that is? That is the most important instrument in your professional career. Your career lives or dies at your keyboard. Maxim number 1. Maxim number 2, learn to write fast and to write well. I can tell you there's never been a paper that was published out of my lab that didn't get rewritten 20 times by me. I tell people in my lab, and I can assure you this is true as an editor, that it's impossible to dress up bad science and make it look good. It's extremely easy to take good science and dress it down and make it look bad. We see it all the time. Please don't exaggerate your work. Please don't overstate it. But, tell a story. If we have time, I can tell you how, I'd be interested how others in the room write their papers. The way I do mine, I can tell you, is the introduction page. I try to make it 3 or 4 paragraphs where you start with a big problem, heart disease, HFpEF, hypothesis, and here we're going to test this hypothesis. So after one page, the reader, whoever that is, an editor or a reviewer or someone who reads the journal, is like, wow, after one page, this is an interesting question, and I want to know what comes next. I'm interested. I believe that this paper that I'm getting ready to read will help me understand this question. And so building a little tension in a story that has a linear theme to it is key in my view. If you're just getting started, number 3, get something out sooner rather than later. Don't try to get a big paper when you're just getting started in academia. Now, this is one of the maxims that goes away after about 5 or 6 years. Once things are moving and you got things going, then you want to pull back and start, making bigger papers for bigger journals. But when you're just getting started, you'll be happier, and your boss will be happier if things are evidence that things are moving. Okay, the publishing process. I'll telescope this. You as an author, and I'm an author, right? You send a paper to the me's of the world. At circulation, we get 15 a day or something like that. And I or one of my senior editors will take a first look at that. And if we think it has legs, we'll send it to expert reviewers. Meanwhile, we will read it ourselves and evaluate it ourselves. And then the reviewers who are experts in this particular type of question will come back to us with an opinion. And we have a meeting in circulation every week. Yesterday morning at 7 o'clock in my office. We do this every week for 2 hours, alternating between 7 a.m. and noon, turns out, because we have editors in 17 different countries around the world. And so we try to make it available and convenient for people around the world. And then the editor gets back to the author with an opinion. The editors make the decision, and not the reviewers. And if there's time, we can discuss about that, more detail if you're interested. Now, we all get bad news. I get my papers rejected. I get my grants rejected. And so what you have to do is take it easy, go home, take a shower, wake up tomorrow morning, and then decide what you're going to do. Don't write a nasty gram to me or to others, in the fit of emotion. Number 4, ask questions that matter. In the world of biology, there's an infinite number of beautiful puzzles to solve. but only a subset of them matter to human beings. For those of you who do basic science, please don't spend your life working on diseases that mice get. Please work on diseases that people get, because you know what? The NIH cares about H. If you want to be funded by the NIH, you better work on human health. I've already told you that one. Number 5, the key to success in your academic career is not papers. It is not papers. It is a theme of observations that build on each other such that you develop a reputation for moving the field forward. You want to be both valid and reliable. Here is reliable, but not valid. Here's valid, but not reliable. Here's neither. And here's what you want. You want both reliable and valid. And the way you get to that point is by doing careful science, careful controls, testing every possible explanation for your observation, and you will ultimately end up here. Maxim number 6. As an editor, I can tell you nobody cares about your fancy paintbrush. We want to know about the masterpiece that you paint with it. You know, we get papers every once in a while. Somebody did a fancy screen, and look, we've got 500 different possible hits. And that, in my mind, is just a 500 different hypotheses. That is a fancy paintbrush. I want to see what you've done with that paintbrush. We don't care about your fancy hammer. We want to see the house you built with that hammer. This is my personal opinion. This is the last one. I personally try to lead in my professional realms by example. That's my own personal opinion. I believe you get more out of your people, and there's more joy in your work if you lead by example rather than by pushing people. That's my own personal opinion. So that's my last maxim. Well, actually, I have one to grow on. My PhD mentor said to me long ago, the key to success is pick yourself up, dust yourself off, and do it again. That is the key to success. There's a Chinese proverb that failure is not falling down, but rather it's refusing to get back up again. Keep at it. Okay, since they're a special audience, I have one more, and this really is my last one. Enjoy the journey. Think about how privileged we are in our profession. When I drive to work, on a Tuesday morning, if I'm going to see patients, they're human beings who entrusted their welfare into my hands. What an incredible privilege. When I'm doing research, I'm trying to make the world a better place for future populations, and they pay me to do this. And furthermore, when I'm doing research, I can use my creative ideas and do whatever I want. I'm like an artist. Think about what a privilege that is compared to most people who have totally noble and worthy jobs, but they're driving a bus or they're selling golf balls or whatever they do. That's wonderful. But look at us. We get to help the world, help individuals, and pursue our creative energies. And that is a wonderful privilege that I sometimes think is easy for us to forget in our world. So with that, I will stop, and thank you for your attention.